Without much further ado, I'd like to introduce Betty Booker. She is a senior scientist in the ACD Bioinformatics team and R&D teams at Advanced Cell Diagnostics. And today she has an overview of probe design technology, application, and new probe requests for you. So with that, I'd like to hand over to Betty. Thank you.
Hi, good morning. Thank you, Rhonda, for the introduction and also for the beautiful rules that have been set for the speaker and also the audience. So we'll go ahead and get started on the presentation. Today we'll review the probe design technology, application, and new probe requests. So we're hoping to help you become familiar with some of our options that we have with our core technology and also how to get started for your own personal probe request. So today we'll start briefly with our core technologies with RNAscope and BaseScope, looking at the assays and platforms that we have available, and then we'll briefly go into some of our applications and give you some ideas about maybe some of the things that you're using in your labs for your experimental approaches, and then move on to some of the target probes that we have available in our catalog here at ACD, which spans over about 26,000 probes, and then also looking at the new probe request form that we have launched last year and gives you an idea of where you can actually start the process when you have a target sequence of interest to submit to our bioinformatics team. And then throughout the presentation, as Rhonda mentioned earlier, we'll have a chance for you to submit any questions you have as I present the information to our panelists who also will answer those questions. But at the end, if we have time, we will also answer some of those questions live. So now moving forward to our core technology, which is RNAscope and BaseScope, we'll briefly start with what our technology has to offer and what RNAscope represents for in situ hybridization. So our RNAscope technology is an in situ hybridization based signal amplification system, which has a unique probe design which enables simultaneous signal amplification and background suppression. This RNA detection in cells and tissue with morphological context at single cell and single molecule resolution. It has three unique features where the single molecule detection from the target probe, the signal amplification as well, which has a branch tree oligo amplification system, and then detection and visualization of your actual target. The assay allows for the spatial mapping of mRNA, link RNA, splice variants, and highly homologous sequences in cells and in intact tissues, all of which can be visualized with either fluorescence or chromogenic labels. The RNAscope technology includes two unique assays to detect RNA, one being RNAscope, and in the tissue context at the single molecule sensitivity with single cell resolution, both based on paired oligonucleotide binding to a sequence to generate signals. RNAscope is our most familiar platform that we have. It is the core technology that we've had, and it's specific for RNAs to target more than 300 bases of your sequence. It comes in one plex, 2-plex, up to about 12-plex for our newer high-plex assay. And this is for research use and also IVD as well. And so these targets, we'll talk more about the actual probe design and also the paired ZZ oligos that you see here. Our next platform is the BaseScope platform, which is for shorter targets. It's about 1 to 3 ZZ pairs compared to RNAscope, which is about 10 to 20 ZZ pairs. This amplification has a much higher amplification, which also allows for us to target shorter sequences such as exon junctions, splice variants, fusion junctions, and also indels. We also can target short sequences such as pre-microRNAs and short guide RNAs, and also highly homologous RNA sequences, and also point mutations, TCR clones, and this comes in either a singleplex or a duplex chromogenic assay format if you're interested in that. So both assays are used in our kits, but RNAscope is for longer targets and BaseScope is for shorter targets. Our technology in terms of the probe design, quality control, and specificity, we have a couple of parameters that we use when we're designing probes, but to be more specific about our ZZ probe pair design that we have, our target binding sequence is the bottom half of the Z that we designed for your target sequence of interest. Then there's a linker region that links the target binding site to the preamplifier binding site, which allows for binding of the amplifier structure. And then what we take into account is what's happening with the actual target binding sequence to make sure that the binding sequence takes into account bioinformatics, also your target and your sample, and also assay conditions. We use these parameters looking at public databases, target species and sample species as mentioned, but we also have where this two adjacent Z pairs two adjacent Z's are required for binding or for the amplifier to bind, and then we run it through our algorithm to provide you with a unique target probe. So we perform these in silico verifications for RNA-scope probes and also for base-scope probes. The probe design algorithm selects the oligos with compatible melting temperatures to provide this unique target probe for your use. There's a verification step at every process here to make sure that your probe is specific to the target. The way we generate signal is dependent on the paired oligonucleotide binding to the target sequence. So this signal is dependent on the paired ZZ oligo binding. And so the paired ZZ oligo binding refers to the Z here and the second Z, which spans about 50 base pairs on your target sequence. This paired ZZ is unique for your actual target sequence, so it should only bind your target sequence. And then the actual next part of the sequence is the preamplifier binding site, which serves as a binding site for the amplifier and the amplification structure that's going to be built. And then we target your particular sequence. The target RNA oligo probe has a full set of spanning across about 1,000 base pairs, which yield about 20 ZZ oligo pairs for your sequence. So your RNA target typically can be longer than 1,000 base pairs, but we target about 1,000 of those base pairs to get you specific signal for your target. More about our amplification system and the signal amplification and how it yields high sensitivity and specificity. The two key features of the RNA-scope technology are the probe design and signal amplification system. The oligonucleotide target-specific probes are depicted as Zs, as mentioned previously, to emphasize the fact that they have two regions linked by a spacer. So the two regions are the target binding site and the preamplifier binding site. The bottom of the Z complements and hybridizes the target transcript and hybridizes to the target transcript, while the top of the Z is the base for the amplification structure here. So this is the preamplifier structure. When the two Zs hybridize in tandem to the target sequence, it creates a binding site upon which a preamplifier can bind and the amplification tree can be built. After the Z pairs hybridize with the target RNA, the preamplifier binds to the top of the Z pair, and each preamplifier can bind multiple amplifiers. So now the amplifiers are also going to bind this preamplifier structure, and each amplifier can further bind multiple labeled probes. Labeled probes contain either a chromogenic here or fluorophore enzyme. This signal amplification strategy yields high sensitivity and allows for visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule that can be quantified. Background is eliminated because the signal is dependent on two ZZs binding next to one another on the target sequence. So if no Z binds here, if only one Z binds, the preamplifier and the amplifier structure will not be built. So that allows us to have the specificity that we're looking for with your target. If both Z's do not bind next to each other, the amplifier cannot be built. So consequently, no amplification will be generated for this signal. Taken together, the combination of the probe design and the signal amplification ensures a high signal-to-noise ratio. The RNA-scope technology has been widely applied for numerous technical and research applications for both academic and biopharma companies. Today, we'll focus on mainly probe design and how to get your target sequence submitted to our bioinformatics team so you can start your experiments with RNA-scope or BaseScope. So the growth and adoption of the RNA technology is best exemplified by the number of peer-reviewed applications that started in 2011. And as you can see in 2020, now we're up to about almost 2,800 publications. And the RNA-scope technology is highly relevant across multiple fields of research, as you can see as well. And so this research includes immunotherapy, neuroscience, infectious disease, inflammation, and also non-coding RNAs. So as a review or summary of the first part of my talk, we want to make sure that you understand that RNA-scope and BaseScope in situ hybridization is for FFP tissues, single molecules, and highly specific. So we focus on our proprietary probe design for the remainder of the talk, and we will talk more about this pair oligo amplification that has no background signal, and the single molecules and single cells, and this targets can be in any species and any RNA. So this in silico validation of highly specific target probes using our proprietary probe design algorithms is one of our main core technologies that we have here at ACD. We would like to further that and talk more about this in the next few slides. So for the next few slides, we'll focus on what we have to offer here. We have about 26,000 probes, and we'll talk about these catalog probes and how to get your probes and your targets submitted to our ACD Bioinformatics team. So as stated, we have about 26,000 probes in our ACD catalog, and we have targeting over 140 species to fit your experimental needs. So one of the main goals when you go to our ACD catalog page, shown at the link here, is to search for your particular target of interest. If you're interested in a gene, let's say like EGFR, you can put that gene information into our catalog search, and all of the probes related to this gene will appear on the search results. Our species range from bacteria, viruses, all the way up to elephants. So species are a wide range, and targets as well. So now more about what we have to offer on our probe pages. So here is an example of the human probe for EGFR. Here is the assay that this probe relates to is RNA-scope. And here is the target probe details here. So the probe name is human HS-EGFR. The gene target details for this particular probe of interest, and the probe name is EGFR, human is the species, the interest ID 1956, and some genes have multiple aliases, so this information should also be available on the probe page. Now, more importantly, the probe target information, that's really important, especially for scientists who are interested in targeting maybe a particular region of a transcript. This information is also available here. Here is the accession number, the target region, targets 505 through 1737, and the number of pairs, similar to what's stated before, our standard pairs are about 20 ZZs, so that is here, and then additional details as well related to the probe. So this probe is for RNA-scope, and this probe shipping temp and storage temp is also provided, and the probe description is here. It's a human probe for EGFR. This is the name of the transcript, and the channel is 1. So in other cases where you may be interested in our BaseScope probes, here's a probe targeting CCL4 of the human transcript, and it's a 1 ZZ probe. So BaseScope probes can be shorter, and so they're usually between 1 and 3 ZZ pairs. So this probe is a 1 ZZ probe, and it's a short target probe. So it's for BaseScope is the assay, and here's the probe name. Similar information is also provided for the BaseScope probes as RNA-scope. Now more information about the target binding details. So each detail here is important if you're interested in targeting a specific region of a transcript. So this region is important because if you're interested in targeting maybe the 5' UTR, the CDS region, or the 3' UTR region, or later, as I'll show by example, maybe you're interested in targeting a knockout region, you must know exactly where this probe binds based on your interest. So here's the transcript number and also the target region and the number of pairs binding. But more importantly, we want to make a note of standard RNA-scope designs use one sequence as an input. However, the probe should cover all NCBI transcript variants. And as an example, here is what we mean when we refer to all transcript variants. So even though this probe targets this particular transcript and is transcript variant 1 for EGFR, as noted by the asterisk, the probe actually covers all of the transcript variants for EGFR, but we only use one transcript variant as an input sequence. So even though all transcript variants are not appearing on the probe design page, for standard RNA-scope probes, all transcript variants are covered based on NCBI transcript variant details. So this probe will cover all of the transcript variants seen here. So more information about the probe human EGFR, additional details. This provides the assay kit compatibility. So what assay can this probe be used with? And also provides temperature and stability conditions, and also provides which transcript variant was used as an input. And more importantly, it provides this information about your probe amplification channel. So this is important when you're interested in using probes in maybe our duplex or our high-plex channels. You need to know as well what channel this probe is designed for. In this case here, the EGFR C2 channel. And so the main difference that you will see in this probe is that the channel has changed. So now the probe amplification channel is C2. And that's also denoted by the C2 at the end of the probe. So our probes come in many different channels. We have now high-plex, which comes in T1 through T2, but our standard RNA-scope probes as well come in C1 through C4. And so here you have C2 for our HS EGFR C2 RNA-scope probe, C1 probe HS EGFR. And if you see, the actual only probes that will have the denotation of the channel will be probes other than C1. So C1 will just be HS EGFR, while C2 will be denoted at the end of the probe. C3 will also be denoted, and C4 will be denoted at the end of the probe name. So if you're interested in channel configurations, your manager will help, your account manager will help you with channel selection. But just to give you an idea about some of our chromogenic readouts, can be either red or brown or red or green. And so keeping in mind that this preamplifier binding site changes based on maybe the channel that you're interested in. The target binding region of the probe is the unique region that will change for each probe that you're interested in, or target region that you're interested in. And the preamplifier and binding site is the modified base channel based on your channel of interest. So from C1, C2, C3, C4, and on up to C5. And this also will be the same for the different channels for our high-plex T1 through T12. So for probe A1, A and C1, the RNA, it targets RNA-A. So this will maybe be used in our duplex assay. And then for probe B and C2, it targets a different gene, which is RNA-B. And then the readout will actually be used in the same assay. So you can use probe A, C1, and probe B, C2 without any cross-reactivity of binding of the two probes in the same assay. So more details about how we name our probes and the nomenclature that we use for our probes. So RNA-scope probes, as mentioned, will provide the probe information, so it's an RNA-scope probe. And then it will provide the species of the probe that the gene targets. And then the gene refers here. And then any specifications that are unique about this probe will also be added to the end of the probe name. And then the amplification channel, if there is other than C1, will be added to the end of the probe name as well. Additionally, our base scope probe nomenclature follows similar, but for base scope probes, we will also include a BA for the platform. And then the species, here we have which represents mus musculus for mouse. And then the gene, NRG, and the subtype This particular probe targets the NRG exon 1, exon 2 exon junction. So I think what most people are here for today is to see what we can target and how we target these regions of interest and give you a highly specific and sensitive probe. So made-to-order probes, we can target tons of different types of mRNAs and RNAs, but some of our common targets that we have are non-coding RNAs, splice junctions, genomic regions, introns, mRNA, unique regions as well, and also short RNAs. Examples of these can be found in our ACD catalog as well, ranging from standard RNA probes like the human LAG3 probes, also standard intronic probes where we can target the HT2 intron 2, and also different pool probes. If you're interested in targeting multiple genes or transcripts, we can target probes, and we can make a pool of probes to target these multiple transcripts with one probe. We also can target different regions of the same transcript for different probes, and those are often identified as iterations of the design. So here's an example of a mouse ISLIR probe 01. So this probe is an iteration of another design. So maybe it targets a different region, or maybe it's a longer probe or a shorter probe, but it gives you an idea that we can have different types of probes targeting different regions, and we can have several probes targeting the same gene in different regions. So we also have a really cool option for a lot of xenograft or PDX models that you may be interested in for your designs. So if you're interested in targeting maybe a human gene in a mouse sample, we have a probe called human PDGFB no cross mouse. So this probe can be used to target a human gene which is being expressed in mouse. And similarly, we also have a probe that can be used to not cross-react, that does cross-react with mouse as well. So if you have highly homologous species, these probes can be used to target the same gene in different species. So here's an example of the rat probe, PDE9A, targeting also the mouse PDE9A gene. So this is an example of when you have highly homologous sequences similar to human and maybe macaca, rat, and mouse. These are the type of genes that you can maybe target with one probe instead of two separate probes because of the high sequence homology. Additionally, for basecode probes, we have a similar setup, but we also have more information provided within the actual probe examples, because basecode targets the junctions, also targets circular RNA, and also targets point mutations and also TCR clones. So we try to give as much information about the particular probe within the probe name. If ever you're not sure about the probe name, you can always contact the ACD Bioinformatics team so we can help you identify which probe will meet your need for your experimental approach. So for example, here is a standard probe targeting mouse EGFR. It's a 3ZZ short target probe. It just targets the EGFR mRNA. Conversely, here is a probe for EGFR 3ZZ short target, but this probe targets a sense probe, and I'll talk more about what a sense probe is on the next slide. But this probe targets the actual reverse complement to the mRNA. So it's not going to target the mRNA itself, but rather the genomic region of this mRNA. And so then next we have probes that can target just maybe a transcript variant that you're interested in. So instead of targeting all transcript variants, as mentioned earlier, we can actually target maybe just one unique transcript variant. Here, this LEPR probe targets only TV1. So it targets only transcript variant 1 without targeting the other transcript variants. But it may not be the case, so we always will check that for you, because it may be specific for only using transcript variant 1, but may also target some of the other predicted models as well, to predicted transcript variants as well. And then next we have target probes that may be targeting several exon junctions, E1, E2, E2, E3, E4, E3, E4. And this is usually denoted as a 3EJ probe. So these types of probes are targeting multiple junctions across a transcript. Next, we also can target a single exon junction, and this is denoted as exon junction 4 and 5. And so this will target the actual junction between these two exons and provide this for FGF8. And similarly, we also have a circular junction that we can target for circular RNAs, and we'll provide some examples for that later in the talk. But all of these examples are essentially ways for us to target and provide specific detection for your gene or transcript of interest. If you're interested in maybe transcript variant 1 or transcript variant 2, we can target those, but we'll definitely confirm them before you order them to make sure that they're targeting exactly what you're expecting for your experimental approach. So more about what's needed for submission, and I also mentioned that we want to make sure it's understood what we're targeting when we design your target probes. So what we're targeting is RNA detection from the endogenous gene transcription. So in gray here, we have mRNA in the gray dotted line. And what we target as a standard antisense probe for RNAscope and BaseScope, we detect the mRNA strand here, which is also akin to the sense strand. So the mRNA and sense strand are similar strands. So we target the mRNA here with the antisense probes. And if you're interested in targeting maybe the genomic region of your mRNA, we can target a sense probe to target the genomic region. So these will be denoted by sense at the end of the name, and these can be done in RNAscope and BaseScope probes. So these probes detect the antisense strand. So instead of detecting the mRNA, it will detect the antisense strand of the genomic region. Our standard probes, as stated again, I probably should say this twice, our standard probes, however, target mRNA or the RNA of your interest. So what is the made-to-order probes and how do you identify these antisense and sense probes? So as stated, the antisense probes are standard. So these probes will be written as RNAscope probe HS-FFR2. This means that this is a human probe for FFR2 mRNA detection. Next, how do you detect the sense probe? So in this case, this is the RNAscope probe. It's HS-FFR2, and it's a sense probe in C2 channel. So this probe will be used to detect the genomic region of this mRNA and does not detect the mRNA of this gene at all, and it's in a C2 channel. So this will be an example of antisense in blue, and in gold, this would be an example of sense probe. So what's needed for your submission to get started? What we need really from you is to provide the exact target sequence of interest. This can be done a couple of ways. We prefer for you to provide either the sequence of your interest or the NCBI session number. The NCBI session number can be provided on the NCBI page. For design, we need the target sequence to get started. The probe will target the submitted sequence. So if you're interested in targeting a particular region or particular sequence, you provide us with a sequence of your interest, and we target that sequence to make our antisense probes. We need the nucleotide sequence of your target, not the protein sequence as well, just as a note. Oftentimes, a lot of our customers are familiar with IHC, and they turn in the protein sequence, but however, we can't use the protein sequence. We actually need the nucleotide sequence of your interest. More importantly, we also need any preferences that you have for your target regions. So for instance, if you're interested in targeting exon 3, please include the exon 3 sequence or the start and stop position of exon 3. Exon number alone is not sufficient for us to design. As we know, exon numbers are not universal across all labs. So a lot of times exon 3 may be exon 2 for some. And so we would like for you to provide the exact exon 3 sequence or the start and stop position according to your sequence. So once you check the catalog probes, then you determine your preferred mRNA or cDNA targets. And then next, we'll get started on if we don't have your gene of interest in our catalog of already these 26,000 probes that we have across 140 species. If we don't have it listed on our website or in our catalog, or the probes don't meet your needs pertaining to the target region, we can provide and start design to manufacture your own in situ hybridization probes known as made-to-order target probes. So with all that said, we want to make sure that if you have a chance to look through our ACT catalog first to determine if any of those probes meet your needs, that's perfect because we want to make sure that we have something to meet your experimental needs to begin with. But if we don't, we can start you in the process to start the NPR of the new probe request. And so the made-to-order probes, go through now a new probe request form. As of last year, we started a new process where all of our probe designs must be submitted through this form. And the next few slides will give you an idea of what this form requires and also the information that can be provided on this form. So the next examples as well will also include examples from xenographs, knockout validations, viral and bacterial targets, codon-optimized sequences, lentiviral constructs, and TCR clones. So to start with the new probe request form, we really start with the customer information because that's one of the more important information that we need on the form. This information is really important because this is how we contact you, and all of this information will be used only to contact you in reference to your probe design submission. So confirming this information is also important to make sure that you've provided us with the correct email, and you should receive a confirmation after your submission, and you will receive contact information from us. If we have any other questions, we'll send you an email with any questions that we have. Moving on to the target information. The information provided here is really important for us to get started. So we provide species of the target gene. So if you're interested in targeting a standard probe, you probably would want to use maybe, for example, maybe a mouse sequence. So you would put that target gene species here of your sequence. And then you put the name of your target gene here. And then if you don't have the accession number, you can actually include the sequence. It's either one. So if you have the accession number, you can include the accession number, but if you don't, you also can include the sequence here. Alternatively, you could also include, if you're doing a xenograph design, the standard designs have the same target and same host species. So the same, if you use mouse, it would also be mouse. But if you're using a xenograph or viral and bacterial design, the species and the target gene and species of the sample should be different. So for example, if you're using mouse here, you would use human here for the sample, if it's a xenograph or PDX model or a bacterial viral target sample. So it just depends on what your submissions are. That information should be put into the input. So for standard probe designs, target gene and species of sample should be the same. So the species of the target gene, species of sample should be the same. You can include the gene, you can include the sequence or the accession number on the left. For more non-standard designs, like a xenograph, you include a different species other than what's found in your target species for your sample. More information that can be included on your new probe request include, is there any additional information that you would like to include on your design information? Is there a specific region of interest that we should target for your design? Is there a specific region we should avoid for your design? And any other information that you should include that will help us design your preferred target probe will also be allowed to be input here into your additional comments or request section. If you would like to check the current catalog with your sequence, you can also include that information here for us as well. So the second half of the NPR is additional information, any non-standard requests that you may have, any questions or any regions that you would like us to include or exclude from the design. That's also really important. And lastly, we also have options for you to submit any files that you would like to provide to us for the design process and feasibility. So you can include the attachments in attachment 1 or attachment 2. And lastly, you also can submit up to 5 requests per NPR. So if you have 5 target genes that you would like to target, you can include 5 of those on one NPR and then submit them all at the same time. So now that we've discussed checking our catalog probes and also determining what region or what target you would like to submit to request, and now the new probe request, now we'll get started on some of the examples. So for our standard probe request for RNA scope, we have here as an example zebrafish, which is Danioreria, and that's the target species, and also the sample species is the zebrafish. And then the gene is PTRX4A. So that should be input here as the gene symbol, and then the accession number is provided in the next input, and then the assay selected as RNAscope. And since this is a standard design, check here if you do not have additional requirements. So this is the box here. An example of how to find the NCBI information. So how do you find the gene symbol and the species and the actual RefSeq information? This information is provided on the NCBI gene page. So if you have the gene ID, you can input that information on NCBI, and that information should be provided. So if you input PTRX4A, the RefSeq information is provided at the bottom of the page. And this is the RefSeq information here. RefSeqs are usually XMs or NMs or XRs for the non-coding RNAs. And for this example, we have a probe in our catalog named DRPTRX4A in the C3 channel. So next, here's a more non-standard design if you're interested in a PDX request. So here is human as a target gene. The species is mouse. This is a xenograft design. So the target sample is, the target gene is human. The actual species sample is mouse. So the customer would like to use a human gene and detect it in a mouse sample. A section number has been provided here. Xenograft for RNAscope. And more importantly, the customer wants to let us know that we should avoid cross-detection of mouse ACE2. We have a probe in our catalog called human ACE2, which does not cross mouse, written as HS ACE2, no cross MM. If you're interested in knockout validations, as mentioned earlier, we have knockout validation options. If you're interested maybe in targeting a specific region, you provide us with that region. This particular customer is interested in targeting the knockout region of 407 through 1214 of this transcript of 6.3. This information is then input on the actual NPR form. This information is here. The accession number is provided. The mouse is mouse 6.3, and please target this region. The accession number is provided, and the start and stop region is provided as well. And this is for a basecode probe here. So this probe targets a 3ZZ span of the 6.3 transcript. So for the knockout validation request, we really recommend that you submit your sequence or your start and stop regions for your knockout validations, and do not select the catalog probes, because catalog probes cover oftentimes upstream and downstream of your knockout regions, and that means that the transcript will still be detected in the assay. Moving on to more basecode probes. So this is the exon junction request with the human MET gene. And so here is a wild type MET transcript. If you see here is a probe, basecode probe for E12, E13, spanning across the E12, E13 junction. And here's a probe for E14, E15, spanning across the E14, E15 junction. And then there's a probe for the E12, E13, spanning across the E13 in the MET delta 14 deletion transcript. And then the last probe here is the E13, E15, which represents the MET delta 14 deletion here. So this probe is in our catalog, but we wanted to show you what is targeted with the exon junction probe. So we're targeting the loss of this exon 14. We would submit this information on the NPR. And this information is really important to submit because we need to know exactly where you would like us to target. On here, you can submit the sequence of this region. And then we would target the junctions at target exon 13 at nucleotide, and then target exon 15 at the nucleotide as well. And so it should not target wild type E13, E14. So the customer wants to let us know that we should not target the wild type E13, E14. And this probe is in our catalog as a basecode probe for human MET E13, E15. And as an example of the images that we have from validations in-house, we have a probe showing that this MET delta 14 with H596 cells. We're showing here with this probe, the E13, E15, which detects the junction. It's showing that it has expression in these cell lines. And so this probe is positive for this particular delta 14 deletion. And conversely, in the E14, E15 with the wild type cell lines, we see here that there's detection in these cells here, while in the actual E13, E15 delta 14 probe, there's no detection because now it's a wild type transcript. And so again, this probe is found in our catalog as a base scope probe for MET E13, E15. So again, here's another example of circular RNA. Junction RNA, it's a similar as an exon junction, but it's a circular RNA junction instead. So if you're interested in detecting maybe a circular junction or back splicing, we can do this as well. Here's a linear example detecting the E2, E3, and the E3, E4 as well. But what happens when you have a back splicing of these two transcripts here? We actually can provide a target probe for you targeting the ASAP1 of the back splicing of the E4, E3. Instead of E3, E4, it'll be E4, E3. And so this sequence can be provided to us. That's very helpful when you provide the sequence of these junctions, so we can make sure we're targeting exactly the junction that you have. And this probe is denoted as a base scope probe, ASAP1, circRNA, E4, E3. So this is a back splicing probe for circular RNA. And so in the comment section, you can include detect back splicing of circular RNA at exon 4 and exon 3. Please see provided sequence. And so again, in the additional comments, target only the circular RNA junction at E4, E3. And then the circular RNA ID also can be provided just for validation of your junction. But here is the sequence that's provided as well. So all this information is necessary for us to start the exon junction probe for the circular RNA. And again, this is found in our catalog as well as a circular RNA junction probe. So moving on to some more probes related to bacteria and viruses. So these can be denoted in the NPR form as bacterial or viruses. We won't have them all listed on the drop-down menu, but you can actually put other and then also select the species which the probe will be used for detection, and you can include that. And if your bacterial or viral species has an accession number, you can include that, or if you prefer to give us the targeting sequence, you also can provide this to us. This example would be for RNA scope as well. And if you're interested in ribosomal RNAs in particular, please include that information in the ribosomal information that you're interested in. Ribosomal RNAs for RNA scope are usually 1 ZZ to 2 ZZ pairs because of high expression. So if you get a probe from us and it's only 1 and 2 ZZs for ribosomal RNA detection, that's because it's high expressors in bacteria. And so here, if you're interested in using a bacterial or viral probe, you also provide any confounding species that you would like us to avoid, or any other closely related species, viral species or bacterial species that you would like us to avoid. For instance, you will put that information here and provide any cross detection or any other accession numbers that you would like us to avoid in the additional comments. Here's an example of a more common target these days. There's a viral RNA target, SARS-CoV-2. So here is an option to target CoV-2 and also target the spike protein of this particular gene. And so here we can target spike S, the spike gene, and then we also need to know which samples you would like to use the probe in. And this is really important because we need to know exactly if you're interested in targeting in this probe in mouse, human, rabbit, ferret, any other species that you may be interested in using our probe in. And so once you have identified your viral target and then you've identified the target region of interest, next you probably want to let us know the host and then also any other species that you would like us to avoid. And this is very common for us, but just let us know exactly what you would like us to avoid, and we can do that with our screen during the design. So next, once you provide that information, you can provide the spiked region, spike S, and then provide the avoid cross detection, and then that information will be provided. mine. And so then that information will be provided on the actual NPR. And if you have other information that you would like us to use, please also provide that information. I'm not sure if the mouse is going crazy. Okay. So next is the codon optimized targets as well. These targets also can be designed with our RNA scope probes. And I'm going to stop for a minute to just restart the presentation to make sure that the presentation isn't skipping.
Hi, Betty, this is Rhonda. Just make sure you've got no animations set up, because if you're in presentation mode, it will automatically forward if you've got any animations on those later slides.
Okay. I think it's just skipping, actually. I don't know. I'm not sure. There's no animations on these 3 slides, actually.
Okay.
So maybe it's just speeding us up for some reason. I'm not sure. I'm just going to, I think we can start at the COVID-19 slide.
Okay, thank you.
Thank you.
Sounds great.
Okay, so I think we can move on to the COVID-19 or the SARS-CoV-2 requests. And those requests are really important just to know exactly what you want to target and what you want to avoid as stated. But we also have new ACD probe sets for the SARS-CoV-2. And those can be found in different probe sets here. And we have 3 sets that are available. And if you're interested in those sets, please let us know. So the next options are vector and lentiviral targets. Those are also targets that you can target with our and provide probes for. If you're interested in targeting a lentiviral or vector construct, please let us know. You can submit those as well. Next is codon optimized targets. These can be targeting synthetic sequences as well. And then the TCR-BCR clonal process. These can be targeted as well. And so for the clonal process, we can target the TCR or BCR clones. And these are, if you have a variable sequence, please submit that with about 70 nucleotides upstream and downstream. And that would be very helpful for our process because we want to target the variable sequence for you. And that's very helpful when we design probes. And here's an example of where our ZZ probes target for these types of designs. And so any repertoires that you have for your TCR, BCR clones, you can upload those as well as an attachment. If you would like us to avoid those during design or detect those during design, just let us know. We can do that for you as well. But those are really important to make sure that you're isolating that particular TCR, BCR clone in your sample. So once your new probe request form has been submitted for a myriad of targets, so from TCR, BCR clones to codon optimized sequences to viral constructs, viral targets, standard probes, or even exon junctions and splice variants. Once we get that submitted from you, we can actually start the feasibility process for the bioinformatics team, and the bioinformatics team will provide you with any information or any questions that we have before we get started to contact you using that information that you provided on the first slide. So once we have that information, we can get started, and you should receive a confirmation of your submission. And once you receive that confirmation, you should know that your probes are now received by our group. And then any additional comments or questions, we'll email you. If we have no comments or concerns about your submission, we'll send you a results page for your probe. So this means that your probe design is feasible for your particular request. For example, here is maybe a zebrafish design for KRT5, and here's the accession number. And the details for the probe design will be provided at the bottom. So here is, I can design a 20ZZ probe design, probe name DRKRT5, targeting 259 to 2185 of this particular transcript variant. So all this information is provided on the results page once we send you that email. And additionally, for maybe base scope probe, here's an example. We will also provide you this information, and any other notes as well we provide on that results email. So once this MPR and your MPR has been completed, your account manager will then assist you with the final stages of coding and ordering of probes. And I think for the overview, we just want to emphasize that specificity is our main goal for our RNAscope and basescope probes. The technology uses a unique probe design strategy to dramatically improve signal-to-noise ratio and sensitivity. The RNAscope technology uses a patented amplification system and fully optimized assay conditions, which add to sensitivity. Multiplexing, the newest assay, RNAscope HighPlex, can target up to 12 targets simultaneously in the tissue at a single cell resolution. TCR and BCR clones, splice variants, and short targets can be targeted by our basescope assay. Again, basescope technology can detect these short sequences and also can target point mutations, additionally with splice variants and your TCR clones. In your new probe request, the RNAscope and basescope probes can be designed and synthesized in 2 weeks for any target in any species. We currently have almost 30,000 target probes on our catalog. So for comments and contact information for our groups, if you have comments or questions related to support in our technology, please contact our support team. If you have questions related to our probe design, you can contact our probe design team as well. And now we'll look at any questions that you may want to send to our team, or any questions that our team, our panelists have already answered. And that concludes the presentation for today.